Thanks, Arijit, for the invitation to this meeting and for organizing more conferences in a year than we do in a lifetime. So, wonderful to be here again, and very nice to see some of the people whose work I've grown up reading and looking at. I see many of them in the audience, so it's an honor to present in front of them. And so let me just get going. So I had to focus on a few algorithms that we've developed to look into microbial communities. Just a quick introduction about our group, because half of my group is here. So if you want to have a chat with them and understand a little bit more about what we do, it will be good. So we are an interdisciplinary group focused on developing various computational tools and scalable algorithms to understand and manipulate biological networks, mostly microbiomes and microbial communities, with a specific focus on metabolic engineering and also health. We also look at performing integrated analysis of various multi-omic data sets. We've looked at large-scale genomic data coming in from coming in from public databases such as the TCGA for cancer genomics and so on. And our ultimate goal is trying to look at impact on the clinic industry and also in the environment. We have a few, you'll see some of the posters that talk about our environmental microbiome initiatives. And we're actively part of several interdisciplinary centers, and most recently, as recent as a couple of weeks ago, we have a new School of Data Science and AI at IIT Madras. So these are the things I want to talk about today. The ones that are highlighted are the ones I'm going to talk about. So few algorithms that focus on various aspects of understanding interactions in communities and with various applications, right? To understand interactions between organisms, to try and understand how the gut recovers following antibiotic treatment, or how we have like some really complex organisms and interactions in extreme environments. So I think I don't need to introduce microbiome to anybody these days because it's become such a commonplace topic. But essentially, microbes thrive in a multi-species dynamic environment wherever they are, and the community structure itself is dictated by a complex web of interactions, where metabolic interactions predominate. And this is so I've been studying metabolic networks for a long time, and so it was easy for me to pivot and look at microbial communities and their interactions in such environments. So it could be the soil, it could be the gut, skin, wherever. And they also have very interesting industrial applications like in metabolic engineering. But across all of these, it's very important to understand their interactions, especially what kind of metabolites are being exchanged and so on. So this is what drives us. So how do we understand microbial interactions in these communities, and can we actually use various kinds of metabolic models? This is the question I'm going to answer today. And you might have seen these kinds of diagrams. So this is a standard chart that you will see in any biochemist's office. And this I always tell the students, is a simplified view of what happens within a cell. Like you have a really complex network out there, and where you have like several molecules that have several different fates in the body depending upon the state of each cell and so on. But it's possible to build these reaction networks and models using various databases today. And ultimately, this is what it reduces to. So it could just be a simple graph of nodes and edges, or it could be like a stoichiometric matrix and so on, depending upon the modeling paradigm that we look at. And what can these genome-scale metabolic models tell us? This is a classic study from over a decade ago now, which only talks about the E. coli model, and it shows how the E. coli model itself has been used for several applications, including metabolic engineering, and most importantly and interestingly for us, interspecies interactions. So how do organisms interact with each other? Can we model them using metabolic models? And there are many other methods to model. So constraint-based and graph-based are the favorites in my group, but there are also population-based methods, agent-based modeling methods, and so on. But there are many challenges in terms of making the models talk to each other, and how do you scale to very large communities and networks and so on. So one classic paradigm is constraint-based modeling, and Sriram here has again contributed quite a bit to this field a long time ago. Wherein it's particularly popular in amongst chemical engineers and so on, where the applications are mostly in metabolic engineering, and you can predict growth rates and the competition between organisms and so on. The other paradigm is based on graph theory, which is essentially comes down to pathfinding in metabolic networks, predicting various kinds of metabolic exchanges and so on. But in other cases, you need methods that are accurate and scalable. This is very important. And we also are looking at methods that can handle the compositional uncertainty. Because sometimes you get only 60 less data where the resolution is poor and you still want to make good use of such data. So, simple stuff here. So how do you convert a metabolic network to a graph? So this is a classic substrate graph where every small molecule is in red. But then you have a small issue here. So you have an edge for glucose to ADP. And if you were to write the whole thing for glycolysis, you'll see that there is an edge to pyruvate as well. And you might erroneously calculate that there is a two-step glycolysis that is possible. In the reaction graph, is not very widely used, so I'll just skip over it. I've mentioned it just for completeness. This is a more useful paradigm. This is called a bipartite graph, meaning there are two sets of nodes where the connections are only between the different sets. There are no links within the same set. So there is no metabolite that directly connects to a metabolite or no reaction that directly connects to a reaction. So ATP and glucose go into reaction 1, or you can think of it as hexokinase, and out come glucose 6-phosphate and ADP, and so on. And when you do this, you have a much better picture of what happens within the cell. You can still draw some erroneous conclusions here if you have to keep a simple pathfinding. But if you start accounting for the presence of all reactants necessary for a reaction and so on, you can get much more accurate predictions of when certain reactions will happen, when reactions won't happen, and what kind of interactions can happen between microbes and so on. So I already mentioned this issue about currency metabolites. So I'll give you a quick overview of some of the algorithms that we've been trying to develop. So the first one is the algorithm called MECTEST, which was developed by my student RP in collaboration with Professor Meghna from computer science. I should already thank all our collaborators because in many of the cases we couldn't have done this kind of work without our collaborators. A, because we had an algorithm which couldn't be implemented without the right data structures that our collaborators would bring, or B, they had like some really exciting data sets which added so much value to the kind of studies that we were doing. So here we use the same bipartite representation I was talking about. So you have the bipartite graph corresponding to organism 1 and that corresponding to organism 2, which is wired through a common exchange medium, common extracellular mass metabolites. So there is this molecule F that moves out. F1 goes out and becomes F outside or whatever. And then F outside again comes into organism 2 and becomes F2. So it's the same metabolite, but it can exist either within the organism 1 or it can be passed on from organism 1 to organism 2. And this will obviously impact our metabolism in different ways and so on. So how do we go about this? We do something, all those computer scientists will recognize this immediately. We do something called a breadth-first search. Only thing it is a little guided. In the sense, it's not a vanilla breadth-first search, but here we take into account the presence of different metabolites before we recognize that a reaction is happening or not, and so on. And that this is ultimately, and then we ultimately assemble the reactions into pathways. You can just quickly download it from the Python package index and start using it. One of the features, it requires only the topology of the reaction network. It doesn't require atom-atom mapping or stoichiometry and so on. It is quite simple and scalable to SF larger reaction networks. And it also efficiently handles cyclic and branched pathways, which are very common in metabolism. And it also looks at alternative routes of conversion, although that's not something we extensively use in this part of the study. So as I said, this is how the models are wired together. And we connect the individual graphs through an extracellular medium, and only the non-common exchange reactions are connected only to the external environment. So what are the inputs to our algorithm? We need the corresponding graphs of whatever organisms are sitting in that system. A set of input metabolites or seed metabolites, this is akin to the medium in which these organisms are expected to grow. And a set of target metabolites. So for the constraint-based folks, this is like the biomass. So essentially, the various sorts of metabolites that a cell has to be able to produce for us to be confident that it is going to grow in that environment. And an integer that bounds the cells with the pathways generated and so on. Okay, so this is very good. This is typically a stumbling block. How do you get the most appropriate medium for growth? And even if you know it, how do you map it to the appropriate metabolites? So this is where we do spend quite a bit of time and effort. We wrap this on various communities. We could even look at communities that are as large as 15,000 nodes and 30,000 edges and so on, where about 1,000 different metabolites could be produced starting from a handful of metabolites. And we were able to identify the exchanges that were commonly seen in literature and so on. But I'll give you some other applications of this methodology shortly. So the next least important question you asked was, now we know that these organisms are interacting, they could be influencing their metabolites and so on. Could we quantify that influence in size? How much does the metabolism of one organism impact that of the other? And we just ask this in terms of, there are some reactions that cannot happen in an organism when it stays by itself. In the presence of a new organism that is healthy, how much of its metabolism gets impacted? So we call it this metabolic separability index, which merely measures the fraction of reactions that are stuck in an organism, which are not relieved in the presence of a new organism. So there are a bunch of reactions that cannot happen in A when it stays by itself, but in the presence of B, about 30% of the reactions are not possible, in which case we call this index like 0.3. Whereas B may not derive the same help, it might in fact derive no help from A. Almost like A might be a parasite on B. So here we say that here, at least according to this arrow, it says that about 1% of B's metabolism is enabled in the presence of A. So this is the kind of measure of the interaction between the two organisms. This is obviously an asymmetric kind of relationship because one organism may be very helpful towards the other, whereas the other may not reciprocate it. If there is no change in metabolism when a new organism is added, then the interaction is zero. It essentially tries to quantify the extent of benefit in terms of metabolism enabled. And we looked at this in several, we had like an experiment of our own and also several other experiments in literature. In all of these cases, we find that the organism that had a higher metabolic separability index was able to grow or outcompete the other organism. Right? It was able to, it had a higher biomass in co-culture. So this is what we used and we've been using to study several different systems. This very simple idea of metabolic separability index is able to capture the interactions between various organisms in different kinds of environments. Overall, our graph models, coupled with some of the constraint-based models as well that we use, can answer several questions. So what are the metabolic capabilities of an individual organism? What are the metabolites that are exchanged between these organisms? How do organisms depend on one another? Do they help each other? Do they actually enable the metabolism of the other and so on? And what are the higher order dependencies, right? Because typically if you see there are these communities where some organisms may seem unimportant. There are some organisms are much more important. How do these communities assemble? Are there like clusters of organisms which help each other more than the others and so on? So all these kinds of questions one can ask and answer with these kinds of models. So again, switch gears and move to another algorithm that we are again developing. The focus of this methodology is to try and leverage 60-less data. So there is tons of 60-less data that's available, but you can't readily use that simply because for most of the modular approaches that we already have, you need more granular information on the kind of genome sequences, the kind of metabolic reactions, and so on. So what we took here is a different approach. So this is a pan-genus metabolic model, and what we do is we build a combined model based on all possible reactions in a genus. So this is just a union, and then we formulate a Balawas objective that captures all of these reactions, and we also add additional parameters to compensate for the variation in the probable variation in the composition in different data sets, in different environments. I'll not get into all the details today. So it just suffices for me to have say that we have a bunch of species that could all be present. So you could have 1, 0, 0, 0, 1, 0 or any other variation in between of these species, and ultimately gives you like one metabolic model that could have different outcomes. immaterial of what is the composition that goes into the metabolic model, it turns out that there are some fluxes that could have a very narrow bandwidth. So this is what we studied. So if you see, there are some metabolites that don't get produced at all in the community. Some metabolites, depending upon the composition, could have a very broad spectrum of variation. Most interestingly, there are some metabolites that could still have a very narrow bandwidth of variation, no matter what is the composition of the community. So this is the sort of idea of, despite the uncertainty in composition, you still have some certainty, at least in certain metabolites, of what is their output. And these are going to be very important for us to understand. So we've studied this for the gut microbiomes and how various short-chain fatty acids, which have chemical importance, are actually, some of them have very narrow bandwidths in certain communities versus others, and so on. Okay, I'm not going through everything, but basically the existing methodology to do this simply launch these models to make PAL models. It doesn't allow for your extra knowledge you may have in terms of certain species. So you may have higher resolutions at some parts of the data and things like that, which you may not be able to incorporate. But our method enables us to incorporate all of these. And these obviously, because of the higher resolution that we have, they tend to outperform conventional genome scale metabolic model-based communities, despite having uncertainty in the taxonomic information that's available. So this is one study, and the most recent study that we completed involves assembling minimal microbiomes for a given data set, for a given environment, which perform a specific function that is desirable to the end user. And this is typically something like you're trying to model a gut. What is a model organ? What is a model gut? What are the fewest organisms that are required in a gut to have a particular short-chain fatty acid profile? I want X amount of butyrate, X amount of propionate, and acetate, and so on. So here what we do is we start with a large microbiome, and we then again use constraint-based modeling here. We reduce the microbiome in steps by removing organisms from this, and especially if it's a large microbiome. Otherwise, we just try to formulate an MILP problem, mixed integer linear program, to minimize the number of organisms that grow into this community, keeping the growth rate as constant as possible. We have certain constraints that can help that, and also keeping the desirable objectives like fatty acid production and so on at a particular threshold. And this can also help us identify the species. Some certain species may most commonly occur in these microbiomes. So are they more important, and do they have significance in terms of the evolutionary trajectories of these species, or if these can be invaded and so on. So all of these questions we can try and start asking using this framework. So here are some examples. So we start with the diagnosed minimal microbiome of size 10, and there is a minimal microbiome of size 3 that is able to show similar behaviors and so on. And many of these organisms that are in these minimal microbiomes have been shown to be important clinically and things like that. But basically, you can see that you can take very large communities and reduce them to smaller communities that have similar function and growth rates. But there are very interesting observations. For example, you may have very different microbiomes that can actually show the same function. We've not had enough examples of this, but theoretically, there's no reason why it's not possible. But we've at least found microbiomes where many of the species are different. I didn't put those figures in here. And again, you can have very many different microbiomes that can show the same function, but still have the same metabolic composition. And this has been a very interesting thing that we noted in microbiome data. When you take, like say, diabetic people or whatever, you will see that there is so much variation in their microbiome. So even in healthy people, you will see that two different healthy people could have almost non-overlapping microbiomes. There are certain species that are conserved, but then you have a large number of species that could still be different and so on. So all these are very interesting things we can dig in these kinds of models. So now I'll quickly go to some of the applications. So this is a particularly good application in collaboration with Murangan Nagarajan from A-Star. This is the pattern that A-Star developed. So what you see here is like a forest fire, and following the forest fire, there's a slow recovery of the ecosystem. And they have likened this to antibiotic administration in the gut. So when you take antibiotics, there is basically a forest fire in the gut which wipes out the diversity of the microbiome to a large extent, and then the microbiome slowly leaps back to normalcy. But it turns out there are about 20 species which accelerate the recovery of the gut. So what is special about these 20 species? Why do these organisms work together? So what we did is we again built these pair-based community models, trying to understand if these organisms have stronger interactions amongst themselves and so on. And it turns out that they work well together. Each of them they collaborate better with each other, these recovery-associated bacteria. And we found that there are certain interactions that are even more interesting. So this Bacteria theater iota micron has a very strong influence on bifidobacterium advancedus, and this was actually tested in a mouse model. I showed that whenever you administrate this probiotic to the organisms, there is faster recovery of the gut microbiome in the mouse. So essentially, the point I'm trying to make here is there is a lot to be understood in terms of the interactions between various microbes and how they can be manipulated to improve health and well-being as well. So in this study, we looked at nearly 50 different species in the gut microbiome. Again, how do they interact? What is the impact of the gut environment? Right. So if you have there is this notion of very high fiber diet or like a high fat diet and so on. So depending upon this diet, you will have differences in how these organisms interact with each other and so on. And again, what happens to the short chain fatty acids and things like that? We looked at 52 commonly occurring microbes and built models and tried to predict interactions. I'll not go into all the details, but this is interesting. So you have bacteria such as Fusobacterium, which derives a lot of benefit from other organisms in one of the media. But when you switch to a rich medium with a high fiber diet, many of the organisms become more independent. But it turns out that there are some interactions that are still conserved in a high fiber diet as well. And this has implications because I think there is some connection between Fusobacterium luteatum and colorectal cancer and so on. And there are these organisms, if they are present in higher quantities or if they are supported by the environment or the food or other organisms, these clinical conditions are less likely to occur and so on. So we're going to switch gears and shift over to the environment. In fact, we'll work all the way out in space to look at the International Space Station microbiome. So this is a very unique system which is under low gravity, high temperatures a lot of time, and of course, the microgravity is one of the biggest stressors there. And this is work in collaboration with Dr. Kasturi Venkateshwaran, who's a really amazing scientist who's generated some exciting data sets of this sort, and again isolated several unique organisms from various extreme environments. And this work was led by Rachita and undergrad in my lab. So what did we do? We looked at 3 different flights, which is the same ISS, but across 3 different time points, and looked at the viable organisms, especially those that coexisted with Klebsiella pneumoniae. And it turns out that this pathogen is present in the International Space Station as well. And we then looked at its interactions and so on, trying to understand both using network-based, graph-based methods, trying to see what organisms coexist with that, and also constraint-based analysis to see what kind of competition might exist between these organisms and so on. And we found various interesting things. For instance, we found that there are certain organisms that are highly dependent on Klebsiella for survival. So if you start targeting Klebsiella, if you start planning disinfection methodologies that will specifically target Klebsiella, you might be able to have a more sterile environment, and especially, you may not want a sterile environment, but you definitely don't want these key pathogens roaming around there. We've also predict several amencilistic and parasitic interactions, And it turns out that one of them we could observe in the lab as well when we tested. So these are electron micrographs that Venkat's group generated. And what we see is that in normal conditions, aspergillus grows nicely, but in microgravity plus the presence of Klebsiella, simulated microgravity plus the presence of Klebsiella, you have a very poor development of the organism. So it's really interesting how in different kinds of stress environments and different kinds of microbial interactions, you observe very different phenotypes for these organisms. So, but I must say here that we always generate far more hypotheses for experimental testing than we are able to test out in the lab. So this is a challenge that we have to get around one day. I hope we will be able to do high throughput experiments at some point of time. But the interesting point is that this helps you ask a lot of questions and get some answers in the direction in terms of possible interactions, possible explanations for why these organisms might coexist or might not coexist, and so on. So with that, I'll head to the last study. I think I still have a few minutes. So this is Pratyay is in the audience and also Shobhan, again in collaboration with Venkat and his group. And here the focus was on enterobacter redundancies and focusing on the differences between the the terrestrial strains and the ISS strains. And it turns out that these ISS strains again were very unique in their interactions, very unique in the kind of genes that they harbored and so on. We did a lot of genomic analysis of these and also looked at their metabolic interactions and coexisting microbes and so on. So it's very interesting again that this organism has a lot of impact on various organisms that it cohabits with and in different environments, right? So in certain, for example, you see that it is present in for some interactions that are present in the first season, right, in the first data set. But when they did this a few months later, those interactions vanished. But then they emerged, some of them emerged again in later months and so on. So you see that even though it's the sealed off system, there is still some interaction with respect to new asteroids going, new payloads and things like that. And maybe different disinfection strategies and so on. And you see that there is so much variation in the microbial composition and interactions and so on. So we identified several important new genes and markers for pathogenic strains about the IS, and especially 14 multi-drug resistant strains were observed there. Almost none of which were found in the terrestrial strains. So this again opens up very interesting avenues for further investigation. So just from simple genomics and some metabolic modeling and so on. This is another interesting study that I haven't had the time to talk about today, but it's a very extreme environment that you see in the hydrothermal vents, where in the deep sea, where you have extreme high low temperatures mixing with extremely high temperature, and so you have a gradient where you have some exotic extremophiles that can grow, and how do they interact with each other? So they have very oversimplified metabolisms and some very unique sulfur metabolic pathways and things like that. Okay, so with that I come to the end of my talk. So some limitations I should importantly emphasize. So most of the graph models that we showed, they only provide a static snapshot of what's happening, right? So graphs are static. We can of course look at various time shots as snapshots and make it a little bit more dynamic. And but still, they're very useful to give you a first cut view of what's happening, and also complemented by constraint-based models which can slightly better try to capture the interactions between or the some the temporal interactions and so on. And again, we generate parallel predictions, then we can validate. So this is obviously something that we continue to look at, and which is where we are looking at collaborators to try and do these experiments to figure out if many of the interactions that we predict happen in different environments and how it is contingent on the environment itself and so on. And the Metcalfe algorithm also is relatively simple. First of all, of course, like all modeling exercises, very contingent on the quality of the input models themselves. And lastly, we look at the simultaneously metabolic lens. I've sort of told you that it's the most important lens to be looking through. But there's obviously a lot more happening in these cells, and we may want to start looking at those as well. Okay. So, anyway, I think I'll skip through this, but just mention this last point that metabolic interactions seem to drive community structure, be it in the gut or be it in extreme environments, hydrothermal vents, and so on. And there are many open questions that if some of you are interested, we can try to address together. What are the key differences between pairs of microbiomes? We are looking at these healthy disease, rural, urban, healthy or bleached corals, and so on. How do these communities assemble? What are the dynamics? How stable are these? And can we manipulate them using either metabolites or organisms themselves? And again, trying to understand the role of these minimal microbiomes, and can we use these to create better model systems to study various environments and so on. And I'd like to acknowledge all my collaborators and students, without which this would not have been possible, and of course our funding agencies as well. Thank you so much. If you have any questions, please do ask right away or do get in touch with me.
Hi. So this would be kind of a different question. So nowadays we see a lot of antibiotic resistance, especially in newborn babies. So the usual method takes 3 to 4 days before the doctors can say this antibiotic is resistant or sensitive. Looking at your metabolic modeling, because 16 days can be done very quickly within a few hours. So if you can use this kind of information, some kind of modeling, what you're doing, and if you can come up very quickly with what kind of antibiotics will be there that this community is sensitive.
I think that's definitely possible. I think it's expensive, so it is not becoming mainstream. And therefore, it's like a chicken and egg problem, right? If you've not done enough effort to get enough evidence and proof, it's not clinically validated, so you don't have regulatory approvals and so on. But otherwise, every time a doctor prescribes an antibiotic, I think it is quite arbitrary, right? And it's typically broad spectrum, because just to maximize the chances of success. The ideal course of treatment to not have resistance is to not have antibiotics, right? You don't put any selection pressure on those microbes at all. But that's not going to be feasible in a lot of scenarios. I really think something like this should be doable, but I think you need to talk to doctors why this is not, because I don't think there are technical restrictions on not being able to do that.
So it's a fantastic talk. I have just one question. So when you say that you have this minimal microbial network, like can you actually somehow extract these microbes and put it on a petri dish and see how they interact, rather than putting it in the kind of, I mean, you said that in mouse model it has been validated and so on. I'm just wondering if that really applies also. Maybe there are other factors and.
Sure. I think typically we use the mouse gut as a mouse is proxy to human, mouse gut is proxy to human gut. So that is why this is typically used. But so in the minimal microbiome, our predictions are mostly theoretical. So what we've done is just validation against other such studies that have reported the presence of certain microbes in literature and so on. But I think it can definitely be put in a petri dish, but you need and these experiments are possible. I've seen people do some of these experiments with there are specific gut-related media that you can use to culture these microbes and study them. Right? And I think definitely that can be done.